Hello everyone and welcome to our webinar on understanding your controls for immunohistochemistry and immunofluorescence. My name is Rebecca Northeast and I'm the technical specialist for Europe. I joined Protein Tech in 2020 following a postdoc in metabolism and a PhD in neuroscience from the University of Manchester. During my time in the lab, I performed many immunohistochemistry and IF experiments, so I hope that my experience combined with some Protein Tech wisdom will help you to get the best from your results. This slide details an overview of what we will cover in this webinar. Firstly, going to go over why do we need to run controls, then highlighting the inherent variability in IHC and IF protocols, how we perform antigen and reagent controls, experimental design best practice, and how to deal with autofluorescence in immunofluorescence experiments. Most of you will probably already know Protein Tech, but if you don't, here's our company introduction. We have been the original manufacturer of antibodies, ELISA kits, and proteins since 2002. We recently became ISO accredited, which means that we can manufacture GMP-grade cytokines and growth factors for use in cell and gene therapies. With 5 global inventory sites, all of our products are in stock and available for next-day delivery. The main difference between Protein Tech and other antibody suppliers is that we manufacture every product ourselves, and you can only buy our products with our label on it. By cutting out the middleman and not allowing anyone else to sell our products under their label, we offer you complete transparency. This means more lot-to-lot consistency because we can guarantee supply in addition to our huge library of original validation data direct from the lab, with complete control over the quality of our antibody, making sure it works in your hands. Protein Tech has polyclonal and monoclonal antibodies against 13,000 targets. Within that, antibodies against 2,500 targets have had their specificity verified by KDK validation. This is the broadest coverage of knockdown, knockout validated antibodies in the antibody industry. This year, we launched correlite fluorescent dye antibodies for use in direct immunofluorescence and protein ladders for use in Western blots. As a company, we measure our success by your scientific success, specifically helping you publish impactful research faster. This graph shows that over our company history, our products are featuring in more and more publications each year. As of today, we have over 70,000 publication citations. Now moving on with the topic for today's webinar. Why are controls so important for antibody applications experiments? Performing controls ensures that your results are biologically real, helps to minimize variables, and enables you to compare data with your own research and that of others. In a paper concerning controls for IHC experiments, the author states, simply put, an immunohistochemical assay that lacks controls cannot be validly interpreted, period. Essentially, this means that due to the nature of IHC experiments, we need to be sure that what we're observing is biologically real, rather than a misinterpretation of artifacts or non-specific binding. During my time in the lab, I encountered these questions to do with my own work and that of others. Questions such as, is this specific staining or just an artifact? Has my antibody not worked, or has my treatment reduced target expression? How can I compare my results and my samples with that of a previous student's or that of a different lab from this cited paper? Is my microscope not working, or is it the reagents, or is it my sample? How do I know what is biological signal or what is autofluorescence? And what are the answers to this? The answer is to perform the appropriate controls in order to understand what your results are showing. Where is the variability in our IHC and IF experiments? Inherent variability occurs through things such as room temperature. So for example, the difference between the room temperature in summer and winter, and this will affect enzyme kinetics for H2O2 blocking, antibody binding, substrate reaction, and chromogen development. Essentially anything that requires an enzyme. Reagents have lot-to-lot variation. Always make sure that you make a note of the lots that you use for each experiment so that you can check back if you get a different result in the future, as this could be due to a lot change. Human error can also cause variability. You get variability in exact incubation times and perhaps changes in application techniques. For example, did you come in later one morning after an overnight incubation? You also get variability through subjective assessments. This is particularly important with experiments that require a subjective assessment for chromogen development, such as DAB, and also in the analysis of results, for example, counting activated cells. You also get variability in pipette error. We add small amounts of antibody each time, so changes here make a big difference in concentration, and always ensure that your pipettes are serviced and calibrated regularly. I'm not going to go over the whole protocol here, but briefly, in immunohistochemistry, the main sources of variability in your results are in steps such as blocking, primary antibody incubation, secondary antibody incubation, and DAB development. These steps contain the most variability through things such as changes in room temperature, incubation times, and buffer composition. And by performing controls, we can help to compare within our own results to account for these variables. For the immunofluorescence protocol, we see variation in similar steps to the IHC protocol as mentioned before. But with IF, we also get added variability due to permeabilization of tissue. For example, changes in the permeabilization time or slight changes in the concentration of our permeabilization agent will control how much antibody can reach intracellular targets. Also with IF, we have fluorophores connected to our secondary antibody. The use of fluorophores conjugated to our secondary will have inherent differences in the amount of light exposure during sample preparation and also microscope analysis, which will in turn affect the brightness of the fluorophores and potentially change results. Now I shall move on to discussing what controls we need to run and why. The first set are our reagent controls. We need to detect that the observed signal is specific for a target, and in order to do that, we can first run a slide which has been processed in exactly the same way, but the primary antibody has been removed from the incubation buffer. Ensure that we still add the other reagents to the buffer and incubate alongside the other slides. Then we will add our secondary antibody as normal and visualize the slides. If you're running a multiplex immunofluorescence experiment, ensure that you do this separately for all your secondaries. This control is highly important and ensures that the observed signal is biologically real for your target, rather than just non-specific staining of your secondary antibody. The endogenous control is where you omit the primary and secondary antibodies from the buffers of both of these steps. Ensure that your otherwise sample is processed in the exactly same way, as this will reveal the level of autofluorescence in your sample, which can easily be mistaken for specific staining. I will later cover how to deal with autofluorescence in this talk. One further reagent control is the isotype control. This is where you replace your primary antibody with that of the identical isotype, such as this one for polyclonal antibodies. The change here is that this antibody is non-immune, and therefore the Fab portion will not bind to any specific epitope in your sample. Ensure that the concentration of your isotype control antibody is the same and is run at the same time as your main experiment. It is also preferable if you buy your isotype control from the same supplier as your primary antibody. This is a more sophisticated version of the no primary control, as it can also account for any Fc-mediated binding by this antibody and any non-specific primary antibody in your sample. In order to ensure that our results are valid, it is highly important to run antigen controls alongside our experiments as well. This is especially important when trialing new antibodies on new cell lines and tissues. Running a sample as a positive control, i.e. one that we know expresses the target antigen, will ensure that our protocol is working and also validates any negative results. You can find potential positive controls through researching the literature or by using overexpressed genetic cell lines for your target protein. Your negative control should contain tissues or samples known not to express the target, such as this cell line which has had siRNA knockdown of HADHA and shows greatly reduced signal for this antibody specific to HADHA. This will reveal false positives or non-specific signal due to non-specific antibody binding. Use UniProt or PaxDB to help find negative controls. In addition, CRISPR-KOs can also be used to knock down your target protein. Where possible, a full set of controls should be run alongside each experiment. However, this is sometimes not possible due to sample limitations or constraints to the number of samples that you can run at once. However, it is most important to run a full set of controls when trialing new sample types, such as a different cell line or tissue, new antibodies or reagents, or a new stock of reagents. If your lot number changes, this is especially important for things such as polyclonal antibodies, as due to the nature of polyclonal antibodies, a different lot will mean that it will recognize a different combination of epitopes and could therefore produce different results. You also need to run controls when rerunning an experiment to increase N numbers, for example. Most importantly, always run your controls at the same time as your main experiment slides. This means that the buffer composition, incubation times, and the room temperature will be exactly the same, and so will remove multiple levels of variability. Here is an example of a slide set of an experiment plus a full set of controls. The main experiment should include all your different experimental groups, however many slides that may be. And your controls should include your antigen controls, therefore a positive and negative target expression sample. And your reagent controls should be that of endogenous tissue where you have no primary and no secondary antibody. Your secondary only controls where you omit the primary antibody but include your secondary antibody, which you run for each fluorophore, as well as your primary antibody isotype controls. If you are pushed for reagents, space in your staining setup, or have limitations due to precious tissue, the two that you must always run are the endogenous and secondary only controls. These will help to account for the majority of non-specific staining and any autofluorescence that you might have in your sample. As stated, always run your controls alongside your main experiment. If you have to perform multiple rounds of staining due to space constraints in your experimental setup, ensure that your experimental groups are split evenly between the rounds. So this is, for example, the best design if you have two groups of experiments. So this here is the best design if you have two groups within your experiment. So for staining round 1, we will have our controls plus an equal mix of participants from group 1 and group 2. And in staining round 2, again an equal mix of group 1 and group 2. This is a poor design because we are not able to state that the results we are seeing when they are split between group 1 and group 2 and separate rounds are due to biologically real data or just due to changes in the inherent variables that you get within each staining round. At Protein Tech, we have recently launched our range of correlate conjugated antibodies. These will help simplify your immunofluorescence protocol as performing direct immunofluorescence with our popular correlate conjugated primary antibodies will reduce the need for secondary antibodies and prevent any non-specific binding in your sample that might occur due to the nature of secondary antibodies. The last topic to discuss today is autofluorescence. Autofluorescence is due to the background fluorescence in a tissue which is not attributed to specific antibody-antigen binding. As you can see in this FFPE, which is formalin-fixed paraffin-embedded human kidney tissue under the green wavelength of 550 nanometers, you can still see some defined biological characteristics in this sample. However, this is just a completely unstained sample. We get autofluorescence due to formalin fixation causing fluorescent aldehyde adducts, red blood cells which contain the fluorescent compound heme, collagen, which is fluorescent as well, and some metabolic compounds such as NADH, which also have natural fluorescence. So how do we fix autofluorescence, or how do we minimize its effects? One way to do this is to always include an endogenous staining control so that when we're looking at our sample, we can tell what is pure autofluorescence and what is biological signal. Most autofluorescence occurs in the green spectrum. So as we can see here in the previous picture to before, when taken under the green filter versus the red filter, you can see a lot more defined biological characteristics under the green filter than under the red filter. Therefore, the autofluorescence is already naturally decreased under this filter. And in keeping with that, the use of red and far red fluorophores, such as the correlate 594 or a correlate 647 fluorophore, will help to minimize this effect straight away. Another way that we can help reduce autofluorescence is to only fix our samples for the minimum time required, and also to perfuse the PBS to remove any red blood cells and the natural fluorescent compound heme. Popular commercial reagents such as TrueView also help reduce any autofluorescence if applied prior to staining. The take-home message from this talk is that controls are vitally important in antibody applications to ensure the validity of our results. Research which controls are best for you and your experiment and that application. Always run controls alongside your experiment. Be critical of your antibody supplier and ensure that they have properly validated the antibody before it reaches your hands and can show their original data. Here are some helpful resources in order to get you finding the best controls for your experiment. So sites such as Uniprot and PaxDB will help you look up protein sequences and also protein expression in different tissues, as well as the Protein Tech website, which contains application-specific tips and tricks and product-specific protocols for you to follow. Thank you for listening. Feel free to contact me on rebecca@ptglab.com. Have a good day.